Hi, as you all know by now, I am Benjamin Beyond for 9D. I will be talking specifically about the DNA replication. Hope you all enjoy it and let's have fun. DNA replication copies the genetic information of a cell. DNA. Replication assures that every cell has a complete set of identical genetic information. Proteins carry out the process of replication. Enzymes help as well. The DNA stores the information only. Some enzymes unzip the double helix to separate strands of DNA. Later, which will be turning out like this basically, and bubbles, and which I'll explain later on. While some does them different, like holding strands serving as templates. The enzymes unzip the double helix by breaking the bases, the four of them, which I will tell later on, and later transforms them like this into two separate strands. A significant enzyme I will use a lot is DNA polymerase. I will tell you more about how this enzyme is going to take place in this replication process. Also, please keep in mind while watching that the blue will represent adenine, the yellow will represent thymine, the black represents guanine, and finally the green representing cytosine. So without further ado, let's get started. The process I'll describe is in eukaryote cells, but the process is similar in prokaryotes as well. Also, let me demonstrate this whole process with my dexterous hands. I'll demonstrate clearly for you guys. These bases are created with colored clay and taped to a red sealed cardboard box, and other materials are color papers like this, for example. Let me give a basic outline first. Number 1, template strands are separated and a replication bubble forms. Bubble. Number 2, DNA polymerase. Well, first, floating faces right here. The DNA polymerase attaches to a separated strand and adds bases. Remember, the green represents cytosine, so it will eventually attach with a black one, which represents guanine. So the base pair of car graph, remember? So it will attach like this, basically. And number 3, 2 DNA results, each having one new strand and one old strand. Thus, when cells' DNA is replicated or copied, 2 complete and identical set of genetic information are produced. Then cell division can occur. Simple? Perhaps we could elaborate on it, right? So why not? As I elucidate, each existing strand of DNA molecule is a template for a new strand. As you can see, the bubble formed right here. Free-flowing nucleotides, which are these, basically, they are floating around. That. Or maybe like that, for example. Anyway, the free floating nucleotides pair one by one with the base on the template strands. They float due to the high concentration and thus stay close together. They add the nucleotides add according to their corresponding base. Take for example the blue one. The blue represents the adenine. Adenine corresponds with thymine, and here is the yellow, so it will go like that. And if you take the green one, which represents cytosine, it has to pair with guanine, which in here is represented with the black, so it's going to go like that. And the yellow, which represents thymine, is going to attach with the blue adenine over here. And last but not least, the black one, which represents guanine, is going to correspond with the yellow one like this, simply. DNA polymerase bond the nucleotide together to form a new strand that are complementary to each template. This process happens rather smoothly and continuously. Replication on other strand is complex due to the chemical nature of the enzymes involving formation of many small DNA segments, and in addition to all this, as the bases match up, the matching happens simultaneously in opposite directions. It opens and adds more, like this and maybe like this. Of course, in reality, there are more than just this bubble. There are multiples all happening at the same time. I have just taken one portion, so it should go like that, 1, 2, and 3. And in the end, 2 identical molecules of DNA result. Each has one new and old strand. So what's going to happen is old strand right here, the new strand, and together like this. So in the end, these 2 identical molecules of DNA result, as I said again. Each has one new and old strand, as I repeat. Thus, DNA replication is known as the semi-conservative, for one old strand is conserved, and while one complementary new strand gets created. There should be in 2 in reality, but I presented 1/2. So it should go like 1, and then 2, old, old, new, and then new. Let me make things clear. Some parts are a little bit vague, so here it is. So what I mean is, in the end, there should be 2 of these, here and there. On the top, old, and then new, and then old, and then new. So there's 2 of them, okay? And what I mean as the DNA polymers say, what I mean is, what they're gonna do is the nucleotides. So it is like this, and then as they do it, as these nucleotides attach, the DNA polymers is going to help attach them together, basically, simply. And not only will this happen in this strand, but as well as in the above strand as well. So basically, while this is happening right here, on the top, in fact, it's going like this as well. So later on, it's like in one point, during the 2, while those bases attach, at one point the DNA polymers will somewhat meet together, basically, and then go in opposite directions again. So like this with the DNA polymer, say, going like this, and on the top, for example, like that, with the DNA polymers is continually going this way. Why not teach you just a little bit more? Let's see. Replication is fast and accurate. About 50 nucleotides are added to every second to a new strand of DNA. Also, the amazing feature is that the fact that it can detect errors, thus replacing the wrong nucleotides. Pretty cool. Replication is happening in your cells right now. Even when you're listening to my maybe excellent or maybe boring video too. DNA is replicated every time your cell replicates. Amazingly, your DNA has replicated billions, if not trillions of times since you grew from one single cell. Human science is not something that to think as easy. It is fun but complex, thus studying hard. Well, thank you for listening to this great video. I will end this with a short music along with the credits. Hopefully I have not all made you into sleep, or hope I elucidated well. I really wish everything about DNA replication is clear right now, right, experts? Well, that's about it. Well, see you and hope everything makes sense.
Well, you done me and you bet I felt it. I tried to beat you, but you're so hot that I melted. I fell right through the cracks. Now I'm trying to get back.